We have to pick a correct choice. Wall hypothesis only indicates how the third base of the anticodon matches the first base of the codon. That's exactly opposite. It needs to be the other way around. The third base of the codon matches the first base of the anticodon. Wall hypothesis states that there should be at least two non-canonical base pairs. Is that correct?
False.
That's false. Remember, there needs to be two canonical base pairs, which are the first two, and the last one, the wall position, is a non-canonical base pair. So B is false. The wall hypothesis states that the non-canonical base pair allows a faster dissociation of tRNA, so I think I forgot to mention this. But one of the advantages of having the third base not binding us specifically is that the bond, the hydrogen bond between that position, between the codon and the anticodon, isn't as strong, which allows for a faster dissociation of the tRNA, which allows for faster translation, so that one's correct. The wall hypothesis is ruled by the recognition of U with C. It's ruled by a lot of different recognition ones, don't worry about that. D might be correct for one base pair, but we saw according to that table there's many, many different combinations. Let's see. True or false, the genetic code should be continuous and non-overlapping. We remember non-overlapping and non-punctuated, therefore it's continuous, so that one is true. The second genetic code refers to the matching of amino acids to ribosomes. So we know amino acids aren't matched to ribosomes. Amino acids are carried by tRNA and matched to mRNA, the codons on mRNA. So we know that one is false. C, usually in great numbers, ribosomes establish 20% of the cell's mass. So that one I think was an italicized note in your PowerPoint. So that one you do need to know that ribosomes can make up 20% of the cell's mass. So, large and small ribosomal subunits are exported to the nucleus. So, we remember that the subunits in general are made up of rRNA, right? And rRNA comes from what? How is rRNA made? It's transcribed like mRNA, correct? And transcription occurs where in the cell? In the nucleus. So yes, that the large and small subunits are exported from the nucleus. mRNA is initially attached to the large ribosomal subunit to start translation. That's false. It's the other way around. It's bonded to the small ribosomal subunit. Despite the larger eukaryotic cytoplasmic ribosomes, eukaryotic and prokaryotic ribosomes may be similar. That is true as well. You remember that the codon is similar between prokaryotes and eukaryotes. The ribosomes need to be similar as well. Even though eukaryotic ribosomes are larger, they operate very similar to prokaryotic ribosomes. In prokaryotic initiation, the thymine is the first amino acid that remains completely stable in a peptide sequence. That is false. I don't think he'll ask you something like that, so if you're studying it, you can erase that one. In prokaryotic initiation, there is an exclusive tRNA that will determine this process.
I don't know what that's saying.
So, remember that there is one tRNA that binds to methionine, but then something else comes and edits that methionine. So depending, I didn't phrase this very well, so we won't ask it like this, but you should know that if he writes it like tRNA bond to methionine F, that is your special tRNA amino acid that starts initiation. If he just mentions a tRNA, you need to know that it's the same tRNA from methionine F and the same tRNA from methionine. So actually, let's look at the other choices. In prokaryotic initiation, once methionine is formulated, the process will stop. That is incorrect. In prokaryotic initiation, tRNA will read a UGA codon. Well, you aren't required to remember the codon, so remember that UGA is a stop codon, so that's not involved in translation. So the most correct answer is B. Which of the following statements is true? The two ribosomal subunits must bind each other before mRNA enters the complex. That is false. Remember that mRNA must first bind to the small subunit by itself. The large subunit will come in later, after the initiation factors are released. A formal transferase removes the formal group in methionine to keep in the peptide. Is the formal transferase the one that removes the formyl or adds the formyl?
It adds it.
Adds it, right. There's a deformylase that removes it. mRNA recognition and alignment involves the small subunit component in the small ribosome. That's true. The Shine-Delgado sequence is known as a pyrimidine-rich sequence at the 3' end. So this is a little bit strange, and there are two lines in your notes where they mention this. The Shine-Delgado sequence is purine-rich at the 3' end, bonding to a ribosomal sequence that is pyrimidine-rich at the 5' end. That is detailed in your notes. Make sure you memorize those two lines as well. Let's see. Regarding the large and small ribosomal subunits of prokaryotes, it can be said that the small subunit needs to be bound by initiation factor proteins to form a complex. We know that does happen in initiation. The large subunit will bind the complex when initiation factors are lost. We remember that the smallest subunit is where the mRNA is initially bound to, and during initiation factors come in. But the large subunit is not allowed to come in until the initiation factors are removed, so that is true as well. Only one of the subunits contains a peptidyltransferase activity. Which one? The large subunit, so it is all of the above. During elongation, 3 initiation factors are required. That is not correct. 3 elongation factors are required. The second tRNA binds to the initiation factor, I think that was supposed to be EF2, that's my bad. EF2 containing GDP. So I'll edit that and put EF2, but is it GDP or GTP? GTP, so that one is incorrect. A peptide bond is formed between 2 amino acids by peptidyltransferase. So we remember that peptidyltransferase is the one that's elongating the peptide sequence by binding the amino acids together, so that was correct. The ribosome moves towards the 5' end of the mRNA 1 codon at a time. That is incorrect. We know mRNA is read from the 5' to 3' end. Which of the following statements is not true? 5 phosphate bonds are used per amino acid during translation. So there's 2 ways they could phrase this. 5 phosphate bonds is correct, but in your notes they also mentioned 3 ATPs. So you need to know both, 3 GTPs, so you need to know both ways they could ask this. 3 GTPs are used per amino acid, but 5 high energy phosphate bonds are used per amino acid. Both are correct. Peptidyltransferase strengthens the binding between the peptide chain and the tRNA during the termination process. Strengthens the binding during termination? No. That doesn't sound correct. That's incorrect because termination you want to dissociate. In eukaryotes, ribosomes have the same sedimentation coefficient as in prokaryotes. So this one is a complicated way of saying a sedimentation coefficient is a way to memorize the size of a ribosome. So if you see the term sedimentation coefficient, eukaryotes have a larger one, prokaryotes have a smaller one, so they are not going to be the same, so A is incorrect. In eukaryotes, the Shine-Delgado sequence allows the ribosome to use many initiation factors. In eukaryotes, while it does use many initiation factors, we know the Shine-Delgado sequence is not involved. There is no Shine-Delgado sequence in eukaryotes. And the last one, in eukaryotes, there's only one release factor. That one is true. In eukaryotes, there's one release factor. In prokaryotes, how many release factors are there? 3. Okay. The other questions on here have to do with the previous PowerPoints, but before we go on to that, let me talk about a few things. So in terms of calculations, from exam 1, he stresses the Henderson-Hasselbalch equation, so you need to be able to solve a question using the Henderson-Hasselbalch equation. You will not be given a formula sheet, so you may need to go back to memorize the Henderson-Hasselbalch equation. Maybe I can type it out. Yeah, so if anyone remembers, it's pH equals pKa plus log of your base concentration over your acid concentration. conjugate base over acid, yes. So conjugate base over acid. So you will be given a question similar to number 2 on the first practice exam. It may even be simpler than number 2. So number 2 is talking about phosphate in water, right? We might remember that there are 3 possible states of phosphate. There's one that has H3PO4, 4 possible states, H3PO4, H2PO4, HPO4, and just PO4 with no hydrogens. So you need to know which one of these pKa's represents a transition from one form of phosphate to another form of phosphate. From H3PO4 to H2PO4, which one of those numbers represents that transition? The 2. So it always goes from lowest to highest. So you need to be able to redraw that diagram that you used to draw all the time in order to solve this question. Notice that specifically they're talking about equal molar amounts of HPO4 and PO4. So which of these pKa's do we need to be using? So that one has to do isoelectric points though, but this one we only need to use the last one, okay? Because we're only talking about one transition between HPO4 and PO4. So if we plug that number in to our Henderson-Hasselbalch equation, it's pH equals pKa plus log of those two concentrations. He will give a question that's pretty easy to calculate. He says it's equal molar amounts, so it should be log of 1 over 1. What is log of 1?
0.
0. So this question is just a way of saying pH equals which correct pKa. The answer should be 12.4. So you will be given a question like that. Please refresh yourselves on the Henderson-Hasselbalch equation.
I'm so mad at this class.
So there's probably other things he's going to ask for exam 1, but again, only 30% of the exam is going to be exam 1 and 2. However, he did mention specifically in class that from exam 2 he's going to ask you about enzyme kinetics. So I'm going to bring that up.
No. That's the way he said. I don't think it should have been.
This is chapter 13a.
It's 2 right here.
You remember this graph?
But it doesn't affect it that much.
Yep.
I didn't get the
Okay. So we're going to talk about enzyme kinetics. He's going to want you to still be able to identify what's competitive, what's non-competitive. He's going to ask what's your Km value, what's your Vmax value. And he might ask you to calculate Kcat, so we're going to go all of that right now. So talking about enzyme kinetics, we know that in general, a Michaelis-Menten enzyme has what 2 sites? An active site and an allosteric site. The active site is where the substrate is going to bind. The allosteric site is where certain regulatory factors are going to bind. If you are told that a competitive inhibitor is added to a solution of an enzyme and substrate, which site is a competitive inhibitor going to bind to? The active site. That's what competitive inhibition means. It's competing with the substrate for the same active site. Non-competitive inhibition, by inference, is going to bond to the other available site, which is the allosteric site. It's not competing with the substrate. You will need to be able to identify the difference between the two on this graph. Suppose it was competitive inhibition. Let's say we have 5 inhibitors and 5 substrates. That means 5 competitive inhibitors are going to have a huge effect on the rate of the reaction. But by increasing the substrate concentration, let's say now you have 1,000 substrate and only 5 inhibitors, we know that the rate of the reaction is going to be very similar to the uninhibited reaction. I remember someone told me in the first exam that there was a question asking about some doctor has a patient that's poisoned on methanol, and she says you treat it by giving him ethanol. That implies that methanol is doing what type of inhibition? Competitive inhibition. Because you're increasing the substrate concentration of something to overcoming inhibition, that only applies to competitive inhibition. Okay? Notice that non-competitive inhibition, when you increase the substrate concentration, you cannot recover Vmax. So you need to be able to identify those factors here. There is another graph that you could give. That's a linear version. Okay. When you're given this graph, you need to be able to calculate / find Km and Vmax. So you may need to, in terms of equations, it's probably best to memorize that the negative inverse of your x-intercept will tell you Km, and the inverse of your y-intercept will tell you Vmax. The only other equation from here that's really important is the standard Michaelis-Menten equation. So that is up here.
I hate this stuff.
Is that one right here. You do need to know that because he may ask you a question using that formula to calculate something. He may give you substrate concentration and ask you what's the velocity. He may give you everything but Vmax and ask you what is Vmax. So make sure you know that formula. You do not need to memorize the inverse version of this formula. The inverse version of the formula, really you can just use the graph to find values. So Km, conceptually, what does Km tell you about the enzyme?
How well they associate.
How well the enzyme and the substrate bind together. So a low Km means that the enzyme and substrate bind very tightly. A high Km means they do not bind as tightly. Vmax tells you the maximum possible velocity, assuming you have all your enzymes filled with substrate. So that's what Vmax tells you. The other thing he could ask you to calculate is Kcat, which is the turnover number, which tells you how efficient an enzyme is. So if he asks you to calculate it, it is Vmax. So if you're given a graph, you need to identify Vmax, divided by the concentration of enzyme, which will have to be given. Okay, so make sure you know that formula as well.
Josh, what is it? Is it when it's low? What is the difference between a low Kcat and a
So the higher a Kcat number is, the more efficient it operates. So conceptually, Kcat tells you if you have a low substrate concentration, which enzyme is going to operate more efficiently. So you want a high Kcat. We know that if you calculate Kcat and it's near 10 to the 8, that is what? Your diffusion constant, right?
So you want a high Kcat and a low Kn.
Yeah, high Kcat and a low Km. And that will give you 10 to the 8. Yeah, a bunch of these are around 10 to the 8. Once you reach 10 to the 8, even if you have a higher number than that, it won't change anything. Once you reach 10 to the 8, you're at maximum efficiency. You're at the diffusion constant. So those are the types of things you could ask those are the type of calculation questions you could ask involving enzyme kinetics. There's a few practice questions he has on exam 2, and he has a separate practice set called enzyme kinetics that you should study as well. Let's see. Okay, so for now, that's all I'm going to go over concerning exam 1 and 2. We need to go back and focus on exam 3 material.
Yep. Yeah, I just did the same thing for my 1 and 2.
Remember that in general, he says he cares about big picture when referring to exam 1 and 2, and he says that structures aren't going to be important for the final. That's what he said in the last class. So we're going to go over DNA replication and transcription, and I think that'll be it. For DNA replication, you need to be able to recognize the difference between prokaryotic and eukaryotic DNA replication. You need to recognize what enzymes are involved and what activities some of those enzymes have. Once again, if you see history stuff, don't worry about it. the replication fork. So you may be given a replication fork or a more simplified version of this and be asked to identify where is DNA polymerase is going to be. Where is DNA polymerase 1 or 3 going to be? Where is the primer going to be? Which direction is replication going to go? Things like that. So we know when DNA is being replicated, what direction is the DNA being generated in? 5 to 3. Therefore, it must be reading the template strand in the 3' to 5' direction. Someone told me in the third exam that there was a question where he gave you an open DNA strand and he asked, label the parts where you're going to find DNA polymerase 1, label the parts where you're going to find DNA polymerase 3, correct? Since we know that DNA polymerase is going to read in the 3' to 5' direction, you must pick the open part of that that had the 3' that is opening in the 3' to 5' direction. There's much simpler pictures of this. I think if you type in replication fork in prokaryotes, you'll see it. But it's not too bad. Notice that if we're talking about prokaryotes, there's one origin of replication. And if you can imagine, prokaryotic DNA is circular, correct? So that means it's opening in both directions on that circular DNA. Okay. So that means on the left side, there is one leading strand and one lagging strand. On the right side, there's another leading strand and another lagging strand. Because you have 2 leading strands, you're going to have 2 places where DNA polymerase 3 is going to be. Because you have 2 lagging strands, you have 2 places where DNA polymerase 1 is going to be and where the primers are going to be. So you need to be able to identify those spots in the replication fork. You probably won't get a replication fork that looks like this. It'll be the much simpler version. You do need to know the different enzyme functions in DNA replication. So in prokaryotes, there are 2 main DNA polymerases, 1 and 3, correct? Which one is your main DNA polymerase in prokaryotes?
3.
DNA poly 3. DNA polymerase 1 is only involved in which strand, the leading strand or the lagging strand? The lagging strand. Its primary function is to remove the primer and fill in that empty space with the rest of the nucleotides. So there's a little bit of a tricky concept that wasn't really detailed in the PowerPoints, but if you look it up in your textbook, they mention this. That DNA polymerase 3 actually generates DNA in both the leading strand and the lagging strand. So hear that DNA polymerase 3 generates DNA in both the leading and lagging strand. However, DNA polymerase 1 is the only one that can remove the primers in the lagging strand and finish off the DNA replication in the lagging strand. Okay, that's DNA polymerase 1. You are also probably going to be asked, what activities does DNA polymerase 1 have? What activities does DNA polymerase 3 have? So we know DNA polymerase 3 is the one generating DNA. It's one of the ones that generates DNA, so it must have a 5' to 3' polymerase, correct? It also needs to be able to edit DNA that it makes, so it has to have some kind of exonuclease, but which one, 3' or 5'? 3' exonuclease is your editing activity. So DNA polymerase 3 has the 5' to 3' polymerase and the 3' exonuclease activity. DNA polymerase 1 is also generating DNA, so we know it has those same 2 activities. But DNA polymerase 1 must remove primers with what activity? 5' exonuclease. So you need to know the difference between polymerase 3 and polymerase 1. In DNA polymerase 1, there's a special fragment you can generate called the clean-out fragment that has only 2 of the 3 activities. And which 2 do we want? The DNA polymerase and the editing function, the 3' exonuclease. So if you see the term clean-out fragment, you know it is DNA polymerase 1 without the 5' exonuclease. So those are terms you need to know. Other enzymes involved in DNA replication, besides the ones that actually generate the DNA, what opens the DNA? Helicase opens the DNA. What does SSB stand for? Single-stranded binding proteins. That's also important. It prevents the single-stranded DNA from rewinding into double-stranded DNA. The beta clamp. Why is that important for DNA polymerase 3? What does the beta clamp help with? Increases processivity. So you need to know what processivity means. It allows DNA polymerase 3 to generate a lot of nucleotides in a row without dissociating from the DNA. That's what they mean by increased processivity, is allowed to go on without dissociating. That's the function of the beta clamp. What about, what connects the Okazaki fragments? The ligase connects the Okazaki fragments. What is needed to generate the primer? Primase. So primase, ligase, helicase, the two DNA polymerases. What is relieving supercoiling?
Gyrase.
Gyrase and topoisomerases, so you need to know that. If you see them phrase it, a little bit strangely where they say gyrase is relieving supercoiling. It does this by either adding or subtracting supercoiling. So that's what they mean by relieving supercoiling. You will either do it by adding or subtracting supercoiling. Let's see, gyrase, okay, so that's, I believe, everything. Let's see.
Need to know any differences between the top isomer and the gyrase, because gyrase is the top isomer.
Yeah, gyrase is a special type of topoisomerase. So in terms of comparing like what you need to memorize and everything, you do need to know what the difference between topoisomerase 1 and 2 is. And that one's fairly easy to memorize. The topoisomerase 1 cuts one DNA strand when it's relieving supercoiling. And topoisomerase 2 cuts 2 strands of DNA when it's relieving supercoiling. Gyrase, you need to know, is ATP dependent, and it unwinds DNA, it relieves supercoiling ahead of the replication fork. Okay, so in talking about DNA replication, prokaryotic termination, we know that prokaryotic DNA is circular, which gives it a very nice advantage that it can finish replication when it reaches the what? What is that point called at the opposite end of the origin of replication? The ter locus, the termination locus. So when it reaches that point, termination occurs partly because what protein is at the ter locus? The TUS protein. So you need to know the TUS protein is bonded to the termination locus, and you need to know what activity the TUS protein has that allows DNA replication to be terminated. Contra-helicase, exactly. So we know that helicase is very important. Helicase is the one that opens DNA ahead of the replication fork so that you can generate DNA. If you have contra-helicase activity, you are preventing the helicase activity, so you're stopping prokaryotic DNA replication. In terms of eukaryotic DNA replication, he doesn't really care too much about the details. He just wants to know some general differences. So for eukaryotic DNA replication, you know that it has many different polymerases involved, from alpha to epsilon. You need to know which one is the main one generating DNA in eukaryotes, which is which one? Delta. So you need to know delta is the main DNA polymerase in eukaryotes. You need to know that's the one that has the highest processivity. Remember that there was well, this isn't the exact diagram I'm looking for. There's a question, a critical thinking question he had, where he gives you a circular piece of DNA, and he inserts a plasmid in that circular piece of DNA, correct? It looked really bad, and it looked complicated, but it's not too bad. If you get those critical thinking type of questions, let me take this chance to point out that you may not be able to solve it on first glance. Finish the rest of the test if you see those critical thinking type questions where you have to do a lot of stuff. Then go back, and if you're not exactly sure what they're asking, take a look at the answers to help you figure out what they're looking for. For the question I'm referring to, they gave you a circular piece of DNA, and they're saying you need to, they kind of imply you're inserting a plasmid sequence into that DNA. And they want to know how many total fragments are you going to get at the end of this. You can figure that out by looking at the answers and noticing some of the answers had a certain number of fragments, and some of the other answers had the other number of fragments. So that question was really just testing you, do you know what an endonuclease is, and do you know what a plasmid is? So as long as you know those two facts that he goes over, you can solve a more complicated type question like that. Again, he may give one or two critical thinking questions like a regular exam. And that's another reason why you should definitely attend the TA's exam review on Saturday as well, because you'll have inside information on the exam. let's see. We already said, again, he doesn't care about the specifics of eukaryotic replication. In general, when he's comparing it, you need to know that which are prokaryotes to eukaryotes has multiple sites, multiple origins of replication. Oh, they're signing for the second? They have to sign in? Okay. All right, there's another round of sign-in sheets going around. Please put your name on those.
Did everyone sign in at least once so far?
Yes.
Yes, signed in.
All right, so there's a second round. Make sure you put your name on the second one as well. Thank you. Okay. So referring to eukaryotic DNA replication, you need to compare a few things to prokaryotic DNA replication. We know prokaryotic DNA is circular, therefore it has only one origin of replication. Eukaryotic DNA is much longer, so it needs to be able to replicate that much more DNA at a faster rate, so that's why it has multiple origins of replication. Okay. What enzyme uses an RNA template to generate a complementary strand of DNA? Telomerase is one type of this. It's more broadly referred to as reverse transcriptase, correct? Reverse transcriptase is the reverse form of transcription. You're going from RNA to DNA. You need to know where you can find reverse transcriptase. You find it in RNA viruses, such as the HIV virus. You find it in telomerase, because we know, what's the function of telomerase?
Telomeres.
It's the one that's generating the telomeres at the end of your chromosomes, at the end of your DNA sequences. We know without telomeres, every time you replicate your DNA, it'll get shorter and shorter, and you'll reach a point called the Hayflick limit. See, these are vocabulary terms, called the Hayflick limit, where DNA replication can no longer occur. So certain cells that replicate a lot must have very good telomerase, so it prevents this shortening of the DNA. And cells like stem cells, cancer cells, these have very active telomerases. Let's see, so that was the
Those are the things that I was hoping would be on the exam. It wasn't on.
What is the letters RT2 is reverse transcriptase 2?
It's just a certain type of reverse transcriptase. You don't have to memorize like what RT2 specifically is, but you do need to know the activities of reverse transcriptase. The main one is to generate DNA from RNA, but you need to know these other two activities listed there. It needs to break that RNA-DNA bond, so that's what they mean when they say RNA's hydrolase activity. And DNA-directed DNA polymerase. So once your reverse transcriptase has generated your single-strand DNA, you can use the cell's own DNA polymerases to generate a complementary strand of DNA. So those three activities go on when reverse transcriptase enters a cell.